Hi, my name is Morgan Kopecki, and today I'm so excited to talk to you about genetic engineering. We've all heard of genetic engineering in Jurassic World or reviving woolly mammoths, but science hasn't gotten quite that far yet. In our body, we have cells, and regardless of cell type, they all have a nucleus with identical DNA. DNA carries personal information, and it is our body's personal set of instructions. So you have a gene that gives you brown eyes. Instead of saying, cells, make this person's eyes brown, DNA has a different communication system called nucleotide base pairs. Instead of demanding brown eyes, a gene would exist that looks more like this. In 2012, when Jennifer Doudna and Emmanuel Charpentier discovered a tool called CRISPR-Cas9, and Cas9 is a molecular pair of scissors. Cas9 finds its location in the genome by using a sequence called guide RNA. Cas9 can now look through the human genome to find a spot that the guide RNA matches. Once it finds a spot, the Cas9 is ready to make a cut. Go back to our cake example to explain how this works. Let's say we want a cake without strawberries, which is step 2. This would mean that our guide RNA would be 2, or the beginning of our step. When the Cas9 scissors find 2 at the beginning of the step, they cut out this step. The step is now not readable and it is skipped. This is similar to a gene knockout, in which the molecular scissors, or Cas9, makes a cut in the gene, and it now is unusable. Now let's say instead of a strawberry cake, we want a blueberry cake. Now that our CRISPR scissors have cut out step 2, we can replace it with the instruction to add blueberries. In the genome, this would be like if the Cas9 went in and made a cut in the genome and added a new gene. Now let's say that we want a cake with both strawberries and blueberries. Our CRISPR-Cas9 scissors would find the spot in our instruction that says add strawberries and make a cut. Now we could make the add blueberry step a part of the recipe. In the genome, this would be like if we saw a regular genome and then CRISPR-Cas9 made a cut and we inserted a new gene in its place without removing any old genes. Scientists are using virus vectors, or what viruses travel in to infect our body, to actually spread CRISPR-Cas9 in these desired genes and get them to as many cells as possible. Other professors are designing cell lines with genomes that have a pre-positioned place to insert the desired gene. What does all of this mean for us as a society? But it does mean curing thousands of genetic-related disorders. In fact, over 6,000 of them. The ethics of genetic engineering are controversial and will be for a long time. But one thing is certain. When we take the time to educate others about it, fear becomes curiosity, and that is when we begin to use genetic engineering to change the world.